Good evening, everyone. On behalf of the Education and Professional Development Committee, I'd like to welcome you all to tonight's webinar entitled NYSCHP Compounding Miniseries Part 1. We are thrilled to have Dr. Mark Klang presenting compounding drugs for administration through feeding tubes, and Mr. Lou Diorio presenting compounding essentials, the new USP 797 pharmaceutical compounding non-sterile preparations. Mark Klang received his PhD in pharmaceutics from the Arnold and Marie Schwartz College of Pharmacy in June of 2008. He is currently the program manager for the Research Pharmacy Core of Sloan Kettering Institute. He is the chairman of the New York State Board of Pharmacy and chairman of the USP Implementation Committee, and has served as a member of the Expert Committee on Compounding for the USP for 10 years. He is an associate editor for JPEN. He was named a fellow of FASPEN in 2020. In this organization, he is a founding member of the Drug Nutrient Interaction Section. He is the current pharmacist at large for the Long Island Society of Parenteral and Enteral Nutrition. He played for many years as a member of the MSKCC Pharmacy Pestles softball team. Lou Diorio is a principal of LDT Health Solutions Incorporated, a medication safety and quality management consulting company with over 75 years of combined pharmacy expertise serving clients internationally. Lou is a graduate of Long Island University's Schwartz College of Pharmacy. He is an adjunct professor of pharmacy practice for the college and a preceptor for pharmacy students. Lou is a member of the college's Alumni Executive Board. Lou regularly lectures at Touro College of Pharmacy, Touro University. He has extensive experience in IV and extemporaneous compounding and lectures, writes, and consults on these topics. Lou lends his expertise to many state boards of pharmacy and is a nationally recognized subject matter expert on compounding, robotics, and automation. Please join me in welcoming our speakers. Before we get started, I'd like to share a few quick points. For those who have technological issues during the presentation, please use the chat option to communicate with the office. Every effort will be made to assist in resolution. For those who have questions that you would like to direct to the speakers, please use the question box or chat option. The CE code will be provided at the end of the program. So without further ado, I'd like to welcome Dr. Klang to begin the presentation.
Thank you. Thank you for that great introduction. This is going to be compounding drugs for administration through feeding tubes. There. Come on, slide. There we go. Learning objectives. You're going to be able to learn a lot of stuff. Okay, we'll skip all of that. One of the things I like to point out, I wrote an article about drugs going through feeding tubes. Got a lot of references in it. Has a lot of studies, and I have to thank a lot of students from Long Island University and St. John's University that contributed to that. My recommendation to you is to read that article. Also, if you're really bored for entertainment, there's a little podcast about how we did this study. This is to show you that this issue is kind of universal, the ideas about drugs going through feeding tubes. This is a study that was done in Japan. More than half the patients who got feeding tubes were unknown to the pharmacy department. Everybody thought that they were doing the appropriate things. 97% of the people were confident that the techniques that they were giving were appropriate and effective, when actually there were errors. There were lots of errors. They mixed meds together. They crushed tablets together. They crushed delayed release products. They didn't dilute thick liquids. There were a lot of issues associated with it. And it's interesting that this, I haven't seen this done in anywhere else, but this is a great idea, is that we do have a lot of problems with drugs going through feeding tubes. When we put in a feeding tube, where are we sticking them? Well, for the most part, a lot of patients get it through the nose. They get longer. When the feeding tube is placed into the nose, it's going to be long, but it's also going to be temporary. If you're going to have a patient's kind of a more permanent tube, that they'll place using an endoscope or a surgery. These tubes are usually shorter, and but they all have the same type of connections. The smaller tubes have fewer clogs and better tolerated, but the small tubes, like placed with an endoscope, etc., these need good muscle support. So the longer the tube, the more potential for clog formation. Let's go on to the next slide. Oh, yeah, I should point out that most tubes are placed into the stomach, nasogastric or J tube. When they're placed into the jejunum, there's a different issue in terms of the pH of the environment. So when you make up drugs going into the jejunum, there's no diluting capacity. When it goes into the stomach, you have a residual volume that's in your stomach. So you can dilute and you can tolerate thick liquids better. You go into the jejunum, you're going to need to dilute the liquids. Now, most people tell me, oh, I already know all about drugs going through feeding tubes. I got a list about them, the do not crush list. And this is often used as a feeding tube reference. It's interesting about this list. I was looking it up. It was originally compiled by John Mitchell, and he passed away in 2014. The list has been updated by certain groups, and it's available through ISMP website. But actually, if you're really looking for an updated version of it, look at the pharmacist letter. They've been updating it now, and they have a 2025 version. But also note that the list includes drugs that taste bad. But we don't really care about drugs that taste bad going through a feeding tube. So therefore, those could be crushed. It also lists drugs that are on the NIOSH hazardous list. Those drugs should also not be an issue. We need to use proper PPE and proper handling of these things, but it does not limit that they can't be administered through a feeding tube. When I talk about feeding tubes, I have to also talk about the food that's going through that feeding tube. Most of the foods have intact protein. That means that they're going to react with acid to form gels. Ensure osmolytes are commercial products. If you give these commercial products or even blenderized foods through into the stomach at less than 50 mLs per minute, you're going to get a retrograde clog. Where you don't get clogs with these types of settings is elemental diets. It's interesting. Elemental diets were actually developed by NASA. They had this idea that well, we couldn't put a toilet bowl on the early space capsules that went into space, and so they made elemental diets. Well, the guys with the right stuff smelled this stuff and said, "Nope, this is the wrong stuff," and so they no longer gave it. And though Norwich Eaton now makes Vivanex, which is an elemental diet, there's also peptide products that also don't react with acid. But these things all have higher osmolarities, so therefore there's a tolerance issue associated with it. And there's also some question whether really is there a need for elemental diets. Well, since we have our bodies can break it up. Oops, I'm sorry, that just went back. I have no idea how that happened, and I hope I never do it again. Oh my gosh. Oh my gosh. I'm sorry. Where did my slides go? Hold on a second. Come on. Where's the slides? All right, here we go. Sorry about that. When we look at oral drug delivery, we look at the drug has to dissolve. When a drug is swallowed orally, it has to be absorbed into not absorbed in the stomach. It has to dissolve in the stomach. It's faster when it's an ionized salt. Weak base drugs also have to dissolve in the stomach. The reason I bring this out is that if you have drugs which require stomach absorption, or not absorption, dissolution, then you will also if you give them into the jejunum, you have to dilute them first. You have to get them into solution beforehand. Absorption is mostly post-pyloric. Very few drugs are absorbed in the stomach. They absorbs the post-pyloric area is around a pH of 6.8, and you require a non-ionized state in order to be absorbed. There is a large surface area of the intestine that may compensate for poor transport, but still it's best to have the drug in solution in order to be absorbed. Formulation issues. A number of drugs have beads and capsules. So what do you use? Do you use water or do you use juice? Well, if it's drug beads that are formulated with an enteric coating, like tamsulosin or lansoprazole, you can put those in with apple juice and it will not clump. It keeps it resistant to the acid. If you mix it with water, you mix these drugs with water, you're going to get a nasty clump. Juice can only be used for gastric administration, can't be used for post-pyloric because it has a high osmolarity. ODT products are really handy for feeding tube purposes. ODTs can be mixed with water, not just desiccant juice, but realize that they are not for post-pyloric administration. A lot of them are enteric-coated. Now, some beads have a sugar core, like Emen, Sporanox. These drugs, in order to make, my recommendation is to use a suspension formulation. Do not compound from the capsule. Another alternative is you could take these drugs and mix them in a syringe. You could open them up, put them in a syringe, and get them to be administered. And that's what we did when Emen was not available in a suspension formulation. Syrups and elixirs, they are thick, they're sticky. They're going to have a high osmolarity. They will form clogs with Ensure. They're very sweet. There's a study done by Cutie and Altman. They showed that the things like ferrous sulfate elixir clog the feeding tubes. Now, if you see excipients, if you look at the ingredients or the excipients that are present, if you see something like povidone in the as one of the excipients, this is a drug that is what's called a coprecipitate. If you want to administer this type of drug through the feeding tube, you got to mix it in a syringe. Don't make a suspension because it will precipitate out. What's the big issue that we're always concerned about crushing drugs is crushing extended release drugs. This is a classic story, Trental, pentoxifylline. This drug, when you crush it up, gives you massive effects very rapidly, nausea, dizziness, vomiting, etc. You can make a suspension out of this, and we just have to administer it more frequently in order to avoid side effects. Here's an interesting change that just happened June 2021. The FDA, I've been bugging the FDA for a long time about administering drugs through feeding tubes. I remember the first time I went before the FDA, I told them, we have an issue. You got drugs going through feeding tubes and you have it interacting with enteral nutrition. And the FDA at that time told me, well, if you want to put drugs through a feeding tube, you have to use a separate feeding tube. Well, they've changed a lot since then. So they came out with this guidance for industries so that now drugs to be approved for enteral feeding tube actually have specific testing that's required. So they require that there's an evaluation of the drugs that bind to the feeding tube material. You have to measure the amount of drug before you put it in the tube and then how much actually gets delivered. Well, I haven't found that many drugs that have that issue. Warfarin, I know. Warfarin adheres to the silicone feeding tubes, especially at acidic pH. Amiodarone also has some plastic adherence. There was a study that claimed that phenytoin suspension did, but I think what was the issue there is that the phenytoin was very, very thick suspension, and that's why it really wasn't being measured well. I did my own studies with phenytoin. I did not find that the drug adheres to the plastic. The FDA also came out with a statement that pure liquids do not require testing. They made the assumption that since they won't cause clogs, so that's really not a problem. One of the things is that they required that the studies for feeding tube must be done with a drug drawn up with a catheter syringe and put in with a catheter type connection for the feeding tube. Unfortunately, few feeding tubes have catheter based connections. You see the illustration on the right. The one on the left, the illustration there shows a catheter type connection, or what they call a legacy, or some people call it a Christmas tree. The ENFit is a new type of connection. That type of connection will not fit the other types, the catheter. There's also no mention of post-pyloric. The assumption by the FDA was that all feeding tubes end in the stomach. as you'll find that's not always the case. Here was a classic story where the FDA got it wrong. And the new drug, this new drug called Syndros, is dronabinol. It was a really good idea because the capsule formulation, Marinol, can't be given through a feeding tube. I mean, it's really impossible. There's some people suggest you could stick a needle into the capsule. I don't know. I stuck the needle into my thumb. It's just not very hard to get the liquid out of a capsule. So they made it into this liquid form, and they actually show that it was safe for feeding tubes. Actually, it was safe only for silicone feeding tubes. Polyethylene glycols type tubes, they dissolve because of the high ethanol concentration of the drug. So that's an issue. I also asked them, can they have a special syringe that comes with the product? So I contacted the company and says, that syringe doesn't fit an NFIT. They says, no, we don't want you to use it with NFIT. We never tested it with that. Also, they can't tell me the osmolarity. So considering it has a lot of alcohol in it, ethanol, I really expect that will be, that the ethanol will have a high osmolarity. So the companies following the FDA guidance for feeding tube. Unfortunately, I don't know any feeding tubes that it can be given through. Here's some other new drugs that have been released for feeding tube using the FDA guidance. There's a pancreatic lipase product, which is really handy because Creon and Zempep, they clog feeding tubes. We actually had a case at our institution where somebody put Creon into the feeding tube, and it just clogged the feeding tube for a patient that had to have it replaced. Viocase has been around for a long time. Viocase is another form of pancreatic lipase, but it is not indicated in the package insert that it is approved for feeding tube, but everybody's been using it that way anyway. And also interesting, the Pertzyme is only good for 14 French feeding tubes. That's pretty big. Most of the feeding tubes are a little bit smaller. There's another drug, carbagaloo, and this drug, it is safe for the feeding tube and can be administered, but they recommend with an oral syringe for the nasogastric. I believe you have to use an NFIT. I don't see any real problems with this one. The other one, the Endaravone oral solution, that one has only been approved for PEG feeding tubes, but it's actually been approved for all different sizes. Well, I want to mention about the Pertzyme. They recommend that you mix the contents with applesauce. I guess you don't use the chunky applesauce with that one, but you have to flush the tube. I like apple juice personally. Applesauce sounds like it's going to be more of a problem. But anyways, it's interesting. Here's another thing. When talking about pancreatic enzymes, so as I mentioned, Viocase is the only product that can be crushed. It's not FDA approved for the feeding tube. They also recommend that it has to be given with a PPI. Creon and Zempep contain enteric-coated beads. They form nasty clogs when they're mixed with water. We've done some studies with using bicarb to remove the enteric coating, but it takes about 30 minutes at 37 degrees. It takes a long time. It really isn't worth it. Why not just crush up the Viocase? There was a dietitian at one of the meetings that suggested that you should mix the pancreatic enzymes directly with the enteral formula. This is not recommended. Aspen doesn't recommend it, but this is to similar to what this other product is. There's a product called Relazorb, and this is a product that delivers enzyme effects. So what happens is they hook this up to the enteral nutrition pump, and they pump the nutrition through, and the nutrition then gets metabolized by the enzymes in the Relazorb capsule. So I contacted the company. I says, so what's in the capsule? Oh, enzymes. How much? What kind? No, we don't have to say that because this is a device and we don't have to reveal the contents. So stay tuned. I have a laboratory. I think I'm going to crack open a package of Relazorb and see what's in it. But in any case, the company says that the product doesn't actually go into the patient. The only product that goes into the patient is the metabolized nutrition product. So remember, this is not an alternative to pancreatic enzymes. It's only lipase, not for it will not break down proteins. So if a patient is a feeding tube, why don't we just give the liquid? Well, first we have to ask the question, is there a liquid formulation? Will it be tolerated at the site of absorption? What is the osmolarity of that product? And also, does it contain sorbitol? Sorbitol is a potent cathartic and is found in most liquid medications. In fact, I remember this one case, Marty Kochever over at Mayo Clinic. He had a patient who had diarrhea, gave the patient Lomotil liquid, and patient had increased incidence of diarrhea. Contacted the pharmacy, what's going on here? Oh, we got a generic form of it. The generic form, the excipient was sorbitol. The Lomotil was causing more diarrhea. They switched from Lomotil, switched to Imodium. There was no problem. Next question, why don't we just crush up an oral tablet or open a capsule? That is a drug, furosemide. Furosemide liquid has such a high osmolarity. But if you take up a furosemide tablet, you crush it up, mix it with water, give that through the feeding tube, very low osmolarity, very well tolerated, very well absorbed. If we crush up a drug, how much fluid do we need to rinse, to mix with, etc.? A lot of these drugs contain a lot of carboxymethylcellulose. So cellulose doesn't dissolve. So a lot of times drugs need a lot of diluents. Acyclovir is 600 milligrams. My gosh, I mixed that up with about 5 cc's of water in a test tube, and it started to grow out of the test tube because it has too much cellulose in it. If we just crush the drug, it's not considered compounding according to 795. But if you mix it with water and you give it, and if you add water and you give it within 4 hours, that can be given. That has no issue. But if you add water and you don't give it within 4 hours, that's compounding. So I know Lou's going to go into a lot of the stuff on 795, but I'm going to steal some of his thunder in advance because I want to talk about some of the stuff. The beyond use date is set by both the chemical and physical assessment. If you want to go beyond 14 days for liquids, you got to have chemical assessment, and the chemical assessment has to be done using a stability indicating assay. I have a machine, I have an HPLC, and I do stability indicating assays. There are very few studies that meet this rigorous criteria. Potency studies is not an acceptable test anymore. Potency study means you measure the concentration in the beginning, you measure it at the end, you don't see a change, stable. Not anymore. You have to actually degrade the drug. And what that means is you have to hit it with acid, base, et cetera, oxygen, and you have to see what types of changes occur, and then see whether or not those changes will interfere with the analysis. So this is what we have to do. Also, compounding has to be done now in a clean area. Gloves and other precautions are recommended. The product should be assessed for bioburden, otherwise known as microbial enumeration, USP 61. And in order to do that test, you have to validate it using a bacteriostasis, fungostasis analysis to determine if the active pharmaceutical ingredient interferes with the assessment of microbial contamination. So if you're going to set beyond 14 days, you also have to have a preservative. Now, OraSweet and OraPlus are great because they already have methylparabens, which can be helpful. But once you add something to those solutions, all bets are off. You have to repeat the assessment of whether or not the preservative is still effective. So in order to do that, you got to do a USP 51 test. There's a lot of work in order to make up a liquid formulation. My recommendation, if the drug can be crushed and mixed with water for feeding tube administration, there's really no reason to make up a liquid. It's sending the patient home is going to be the issue. Now, this is way off my feeding tube issue, but I have to mention it when I talk about 795. Don't add flavors. Flavoring is considered a food. There is no uniformity in terms of flavoring. Flavors changed in chemical content without notification. We actually had an incident where that was occurring. I contacted the company. I says, what happened to the, I was mixing Crystal Light iced tea mix, and we were using that as a diluent for gas review, and it was working very well. The company changed the content of the product. Drug precipitated. Flavoring agents do not have stability or compatibility data. When I contact the Flavor RX, they says, "Oh no, our product is natural." Okay, what does that mean in terms of stability? Walgreens, I had a meeting. Walgreens told me that they're no longer going to be adding flavoring because it's too risky. Okay. I talk about cellulose, and there is a lot of cellulose. So look at those screens down below. What I did is I crushed up the drug and I put it through a 100 micron screen to see whether or not it would accumulate on the screen. And sure enough, drugs that have a high amount of cellulose present, they're going to accumulate on the screen, and they're going to get and they're going to clog the feeding tubes. Acyclovir, Lipitor, these drugs all have a high amount of excipients. And I mentioned before, drugs that have ingredients, coprecipitates like povidone. Mix these drugs in a syringe, not separately in a bottle, because you put it in a bottle, it's going to precipitate out. There was one drug was itraconazole. They actually mixed that in a solution, and they measured the levels in the patient. Patient had zero levels. So if you're going to mix it beforehand, put it in a syringe, mix it, make it into a slurry, and give it immediately. RX List is a great source for drug solubility and ingredients. Here's some drugs that really clog up things. Yeah, some of the newer drugs are thick as a brick. They just clog up there. Yeah, they get very thick and they accumulate onto the screen. Capsules, oh my gosh, capsules do not dissolve. And so we find a lot of problems with trying to administer capsules for a feeding tube. Putting in a syringe does not really work out as well. When you're crushing tablets, obviously we want to use immediate release tablets. I usually tell people if it's scored, if it can be cut, usually it can be crushed. You got to be careful with that. Toprol XL is actually an extended release product. Chewable tablets crushed coarsely. What's a phenytoin? Phenytoin tablets actually have a lot of problems with clogging feeding tubes. And coatings form gummy solids. I tell the story often of the patient got an order for amitriptyline 30 milligrams. Why they ever did 30 milligrams, I'll never know. But they had 30 milligrams. Somebody gave 3 10 milligram tablets. These drugs have a coating on them. And when you mix them with water, they actually they will clog up the feeding tube. Shards will also form coated tablets. Lipitor, I find it's easier just to mix the Lipitor with water rather than trying to crush it up, because crushing it actually becomes more of a problem. So we're ready for our polling question. I look to the moderator. A valid remedy for a clogged feeding tube due to an undissolved weak basic drug. It's best to rinse with. Pick your choices. Do we get to play the Jeopardy theme song while we do this? Okay. It says I can't vote. Wow. I'm so happy. Yes. Yes. VioCase will not work to unclog a feeding tube that is due to a drug. VioCase will actually unclog things that are protein-based types of products. And somebody just, I think, hit the crayon because they're just trying to mess with me. And hydrochloric acid is not a good idea. I hate when that goes through the feeding tube. Oops, I hit my wheel. that's what happened. All right, why do feeding tubes clog? Because we didn't rinse adequately. Rinse a lot with purified water. There was an earlier statement from the FASPEN organization suggesting that you need to use sterile water for irrigation. It's not necessary that the water be sterile. It's just GI tract is not sterile. There's actually a study coming out of my place where somebody used tap water. I don't recommend tap water. Purified water is better because some drugs react with the metals that are present in tap water, especially our good old New York water has a high concentrations of calcium and magnesium, and so they probably will interact with some drugs. Drugs have to dissolve to be absorbed, and if there's coagulation of intact protein by acids, and that can also cause a clog. The pancreatic, I love the way to do this. My recommendation is use BioCase to unclog the feeding tube, and I also point out that BioCase actually has some residual inside of it as well, so it can make the clog worse if it's not a protein-based clog because there's a gummy residue inside. But one of the options is you can crush the drug, mix it with 5 mL of bicarb injection, or mix with equivalent of about 1 teaspoon of bicarb powder. Or another option is just crush a 650 milligram tablet. And this needs to be administered through the feeding tube. One of the problems with administering these products into a feeding tube is that you need to get to the site of where the clog is. And that's often a difficult situation. We did some studies with this. We actually used ZenPep because the BioCase wasn't available at that time. We found that the fastest way to unclog the feeding tubes, warm water. Warm water was the best way to unclog the feeding tubes when it was even with a protein-based clog. But if that doesn't work, pancreatic enzymes do work. Clog zapper. We're actually looking at a trial at my institution looking at clog zapper. It uses papain, which breaks down proteins, and it also contains the cellulose to break down cellulose fiber. We're going to see whether or not it stands up against BioCase. But probably the fastest, most reliable method out there is a device called Tube Clear. It unfortunately requires a special introducer, and you got to use a different size introducer depending upon what is the size of the feeding tube. It's also the most expensive device. But if you're looking for something that's going to unclog feeding tubes very rapidly, that might be one way to do it. Some people found it to be too complicated to use. Okay. Didn't we already do this one? That's polling question 1. Yeah. I'm sorry, can we skip ahead? I don't know how this came up here. I thought this should be. We go ahead. There we go. Osmolarity of liquid medications. Actually, there was original for compatibility of medication. It was really studied by Cutie and Altman back when I was at Long Island University. But at that time it was called Brooklyn College of Pharmacy. Dr. Anthony Cutie and Esther Altman, they did a study looking at compatibility, and they found that ferrous sulfate really formed a pretty solid mass. I repeated that study back in here in 2013. I also checked the osmolarity, checked for compatibility. Basically, if the liquid is thick and highly reactive, it's gonna react and form nasty clogs. Furosemide, calcium salts, acyclovir. These products all have very high osmolarities. It's probably easier just to crush the drug and mix it with water. With the acyclovir, I recommend you use the 200 milligram size. The higher milligram size contain povidone and may not dissolve as well. This is a excerpt of some of the studies. As you can see, drugs, not always do the drugs that interact to form incompatibilities are acidic. Some are just had nature of the formulations. And we found that many drugs had high osmolarities that could be really significant. When we look at osmotic issues, a lot of times we say that, is the patient had diarrhea? Is it caused by the nutrition? A lot of times it's caused by the drugs because of the excipients that are used. Study done by EADS found that 40% of the diarrhea was caused by the excipients. You found a lot of patients were getting sorbitol-containing liquids. Try to avoid using sorbitol-containing liquids when you're giving drugs through the feeding tubes. Osmolarity of crushed medication is very low. So all the liquid medications I measured had a greater than 1,000 milliosmoles per liter. Some of the crushed medications actually had pretty high osmolarity, and Mesna, metformin, and calcium acetate all use high osmolarity when I crush them up. I recommend with these drugs you give them with extra fluid in order to improve tolerability. Here's our polling question number 2. Dr. Bauer, he was the guy who first discovered the idea that phenytoin has an interaction with nutrition. He found that the patient levels were very low when they got to suspension formulation through the feeding tube. He advised that the nutrition formula should be held for 2 hours before and 2 hours after administration to avoid the interaction. He explained that the drug is strongly bound to protein in the enteral nutrition, and the patients who get enteral nutrition are prone to diarrhea. What was the mistake that Dr. Bauer made? I open it up top. Okay. The poll is now open. Actually, no, it wasn't the high sorbitol content. He assumed that all enteral patients have diarrhea. Patients who get enteral nutrition are constipated. And heck, if you're a patient with Sloan Kettering, we got a lot of pain meds going on there. Oh, geez, those poor patients are constipated even more. The issue, Dr. Bauer assumed that when you give a liquid nutrition, he actually wrote to me, he says, patients, it's liquid in, liquid out. No, that's not the case. If just because you're eating liquid nutrition doesn't mean you get diarrhea. If you do have diarrhea, let's find out why you have diarrhea. Maybe the drugs or the sorbitol is causing it. There's been numerous studies that people should be withholding tube feeding for warfarin, phenytoin, fluoroquinolone, thyroid, levodopa. Most of these studies were faulty, exception of levodopa. Levodopa actually competes with the amount of protein that you're giving. And if you could time your nutrition around when you give levodopa, you can actually improve the levodopa levels. A lot of studies say that the drugs bind to protein and will cause an interaction. Yes, these drugs do bind to the protein, but the protein is getting absorbed. Phenytoin suspension is very thick. If you dilute it with water, it will avoid the issue. Yes, it binds to the nutrition, but the nutrition gets absorbed. Warfarin actually adheres to the plastic of the feeding tube. It also binds to the protein and the nutrition, but it competes with that to the tube, but the nutrition gets absorbed. Ciprofloxacin suspension actually says in the package insert, don't give it through the feeding tube. What you can do, you say ciprofloxacin tablets and crush them up. Studies by, I forgot its name. There were studies that were done. Yuck did the studies on ciprofloxacin. He actually found that there was no problem giving it through feeding tube. He actually found that ciprofloxacin actually degrades a little in the stomach acid. Thyroid medications are real problematics. The issues always with, as anyone knows, Synthroid, Levothyroid, all these generics, anything with thyroid medication have difficulty being absorbed uniformly. My recommendation, give whatever you're giving the same way all the time, and that way you avoid any issues. Osmotic issues, OraSweet contains a lot of sorbitol. Watch out for electrolytes. If you give an order for 60 MEQs of KCl, you're going to require 6 to 8 ounces of water per 20 mLs. The FDA considers sorbitol a GRAS, generally recognized as safe, but it's more potent and cathartic than lactulose. And when pharmacies compound liquids, in many cases, they have osmolarities greater than 1,000 milliosmol. And remember that the GI tract is only around 300 milliosmol. In the stomach, the stomach allows for the tolerance of up to about maybe 600. The jejunum is very acutely sensitive, no diluting capacity. It's interesting. FDA rules. The generic formulation does not have the same formulation as the brand, so there may be sorbitol may be present in the generic, but not present in the brand. All right, this is getting repeated here. I apologize for that repeat. PPIs, open the capsule, mix it with juice. About 60 ml. Study done by Chen showed that he had to you mix the capsule, you mix it with apple juice, you rinse it with 30 mLs before and after, and you can mix that through the feeding tube. Another alternative, take a PPI, mix it with sodium bicarbonate. That way you can give it either gastric or jejunal. Another option, use the orally disintegrating tablet. We actually had an incident here. Somebody put the ODT and they assumed that because it was placed under the tongue, that it was absorbed in the mouth. It is not. It actually absorbed in the gastric tract, and so it needs to have intact GI tract. Teva actually had an incident. The FDA pointed out that Teva's version of ODT actually clogged and caused a bit of problems. But mixing the oral capsule with water will clog, but if you apple juice is better to mix ODTs. Proton pump inhibitors are frequently a cause of clogged feeding tubes because they require acidic juice for administration. You mix it with bicarb, you can remove the interior coating. That might be an alternative. You give the product as an ODT, might be the best way to do it. I'm sorry. When you give something past the pylorus, it's sensitive to osmolarity. You have to dilute liquid medications in that setting. Smaller feeding tubes are usually used into the jejunum, and so they're also more prone to clogging. One of the issues we came across, enalapril. Enalapril is a prodrug. This needs to be exposed to acid for absorption. Other drugs that need acid for absorption, diphenhydramine, epinephrine, doxycycline. These drugs should not be administered directly into the jejunum unless they dissolve. Some drugs degrade in the stomach. Actually, ciprofloxacin degrades in the stomach, but it actually dissolves better. Are there drugs that should not be administered directly into the jejunum? Some drugs require the acid content of the drug in order to be absorbed. And I don't want to try to pronounce these because I'm going to screw them up. Some drugs require dissolution in acid in order to be absorbed in the jejunum. Ketoconazole, lithium, pravastatin. These drugs require acid dissolution. Actually, I actually got a couple of notices where we found that the antacids were being given into the jejunum. This is not valid. There's no reason to give an antacid. Sucralfate should not be given into past the pylorus. This is my favorite toy. We're using it now with Sloan Kettering. We use the RX Crush. It's a very handy device. One of the things we're looking at is maybe just crushing up the drug and not adding the water until it's ready to be used. And that way we can have the drug administered at the bedside. Another option is putting the drug inside of a capsule, especially when you're dealing with chemotherapy. This is a good way to keep the drug contained. The RX Crush is also very good for chemotherapy. It keeps the pouch does not open, and we found it very successful. We actually did a study using coffee. We put coffee into the drug, and we couldn't smell the coffee when we put it in there and we crushed it up. Very, very handy for that type of use. General considerations. To avoid clogging the tube, yes, using the liquid formulation seems like an obvious remedy. Unfortunately, few drugs are available in liquid, and not all liquids are ideal for the feeding tube route. Consider this: the better the palatability, probably the less appropriate it is for the feeding tube. Putting a drug in a feeding tube limits its dissolution. So therefore, you better dissolve the drug before you put it into the feeding tube. Delivery at different GI sites will alter the amount that actually absorbs. Consider how much drug adheres to the plastic, maybe to protein, and possibly to chelate to metals. Actually, some drugs also require specific GI sites, enzymes, or lipid involvement. So keep that in mind when you're administering the drugs. So generally, oral drugs for feeding tube, if it's a plain tablet, we crush and flush. That's the best idea. Hard powder filled capsules, open them up, mix them, flush them down the feeding tube. Soft gelatin capsules, there's theories that you can actually create a pinhole and actually squeeze out the amount. We found it didn't really work very well. Nifedipine capsules, we actually tried to get the contents out. It was very unreliable in terms of trying to measure the drugs. One of the other issues, we had a lot of issues with this drug, magnesium oxide. We had one clinical pharmacist went around adding, we have a lot of patients that have magnesium deficiencies because of the chemotherapy that they get. And so we usually have many times a magnesium wasting syndrome in cancer patients. And so we went around suggesting everybody use magnesium oxide. And it's great. I mean, magnesium oxide's got a huge amount of magnesium content. Unfortunately, it's worthless. It doesn't dissolve. And since this doesn't dissolve, it's going to clog feeding tubes. Please do not use magnesium oxide. Magnesium is very difficult to replace. And if you want, that's a whole nother lecture on how to get magnesium. Many drugs have poor absorption to solution. Excipients are added to enhance the solution. Excipients are often insoluble in water, as I mentioned, the cellulose. When a liquid formulation might seem ideal for the feeding tubes, remember that no liquids are designed specifically for the feeding tube route. A lot of times liquid meds are for pediatrics or for people with difficulty swallowing. So as dissolution decreases, as viscosity increases, and it also improves the taste. And there are my references. And if you have any questions, we're ready. My slides have self-assessment questions at the end, but we don't have a mechanism to use those. So I'm open for questions. I'm going to hit the stop share.
Mark, it looks like there's a question in the Q&A box. Any thoughts on using soda, for example, Coca-Cola, to clear clogs?
Oh my gosh, there was a great study by my card. We're looking at all different types of remedies for unclogging feeding tubes. Sodas all have acidic vehicles. And so therefore, if they come in contact with enteral nutrition, they will actually make the clog worse. The recommendation in this type of setting, there's a suggestion maybe because it's a weak base, that's what forms a clog. My recommendation is always warm water. There's a lot of nurses I know, I've seen Mountain Dew being used and other stuff of that nature. Not a good idea.
Right, that's good. If anyone else has any other questions, please feel free to add them into the Q&A box and we'll come back to them at the end. And now we can transition into the second portion of our presentation.
All righty. Okay. All right, I think that's working, Gina. How's that look?
Yeah, I can see it.
Okay, perfect. Thank you. All right, so thank you, Mark. Very informative, very scary. Um, didn't realize the complexity that I had walked away from when I left hospital practice. But so now we'll transition and we'll talk about, we'll give you a good overview of non-sterile pharmaceutical compounding. And let's see, with that, okay, so you guys can read the session objectives. We're going to talk a little bit about the activity of water. We're going to talk about the difference between QA and QC. And yes, there is a difference. We're going to talk about training, and we're going to talk about what constitutes a proper master formula record. And for the technicians in the house tonight, welcome. And we're also going to talk about proper garbing in addition to the topics for pharmacists, as well as what Mark pointed out some physical attributes for non-sterile compounding areas. We're going to go into a little more detail, and I'm going to try to give our technician friends some tangible takeaways to add to your compliance plan. So with that, I am going to dive in. Please feel free to put questions into the chat or into the QA function as we go along. And as Gina and Rebecca said earlier, if we do get questions that are that intense or that specific, we're committed to answering them after the session concludes. So not to worry, all your questions will be answered. So with that, I will dive in. I want to point out that with the launch of the revised chapters for not only sterile compounding, non-sterile compounding, the handling of hazardous drugs, as well as radiopharmaceuticals, the USP achieved two of their sort of side goals, and that is to sort of harmonize the format and the form of each chapter. So you need to be aware that the revised chapters are descriptive and prescriptive, but if you're looking for a very specific type of answer, you may be caught in a seam between two chapters. So you may not get a complete answer if you're referring to only one of those chapters. So for example, if your question revolves around a non-sterile hazardous preparation, both a complete answer may reside somewhere in the interface between 795 and 800. So please be aware of that. The other thing that you should really be cognizant of is the use of shoulds and shalls and musts in the chapter. So a should is a recommendation. It is a very strong recommendation, but a recommendation nevertheless. And the shalls and musts are required. So please be aware of that as you navigate through the chapters. All right. So 30,000 foot view. I always speak for those of you that have heard me speak before. I apologize in advance. For those of you that haven't, I always try to put these talks into the context of the three. I like to call them the three Ps. The people, the places, and the practices that will get you to compounding compliance. So the people, that refers in my head to training. The places, that refers to the environments and the physical locations. And then the practices are just that, the policy and procedure and the other background documents. So really, as we go through the slides this evening, please try to put it into those buckets, and I think you'll come out the other end of the tunnel in much better shape. So USP 795, the chapter includes, as far as dosage forms, but are not limited to, obviously, solid orals, liquid orals, rectal preps, which was a change from the last iteration of 797, vaginal preps, same way, creams, gels, ointments, and nasal and sinus preps intended for local, applications, so sprays and nasal irrigations. And then obviously otic preps with the caveat that if they're used in the presence of perforated eardrums, obviously they must be sterile. So just be aware of that. Talk for a second about the scope of the chapter. Please be aware that this is not a pharmacy-only problem. The chapter, the folks that are covered under the USP in this context are not only pharmacists and technicians, but nurses, physicians, veterinarians, chiropractors, naturopaths, and any other healthcare professional. So it is universal and needs to be applied in all practice context. And we are seeing, believe it or not, we are seeing actual policing by not only local health authorities, the Board of Pharmacy, but also the FDA themselves. So non-sterile compounding is not for breakfast anymore, children, as the TV commercial goes. The compounding facilities leadership and all people involved in the preparing, storing, packaging are responsible to make sure that the applicable practices and quality standards are in place so that you can control the process and you can have a constant and level playing field, making sure that the consistency between the formulations, whether they were made on Monday by pharmacist A or on Friday by technician B, are all the same. So please be aware of that. And then personnel engaged in the compounding and dispensing of non-sterile preps must, in addition to the USP chapter, must comply with all state, local statutes, rules, and regulations. So with all due respect to Dr. Klang, please do not forget about the State Practice Act. Right? And nothing that I'll say tonight flies in the face of that. If it appears to do that, that is not my intent. Please start with the State Practice Act and then work USP into your compliance plan, please. All right. These are the exclusions to the chapter. The following are not considered compounding and not required to fulfill the requirements of this chapter. Dr. Klang mentioned one of them. This is just so in brief, non-sterile radiopharmaceuticals. For those of you that are in that specialty or subspecialty, please refer to USP 825. Reconstitution of a conventionally available formulation is not considered compounding. If the instructions for reconstitution are in the package insert, you are not compounding. Repackaging is governed by the New York State pharmacy regs covering repackaging, as well as USP 1178 for further recommendations. Obviously, splitting or breaking or cutting tablets that are allowed to be to do so, that is not compounding. So for our brethren, primarily in long-term care, that is a welcome relief. And then the last one is where some confusion pops up from now and again, is what is considered administration. So the prep of a single dose for a single patient when administering, and that administration begins within 4 hours. I think Mark gave a great example during his talk about the difference between a single dose given within that 4-hour window and preparing a batch or a collection of doses. That's a separate issue. That would not be administration. So crushing a tablet or opening a capsule and mixing with food bedside does not constitute compounding. So let's be real clear about that. All right. So here on the next 2 slides are really the cliff notes of my talk. I condensed all of the major revisions into what I think is a more digestible format. So a major change is the identification of a most responsible person for non-sterile compounding. Now, for those of you that have jumped on the bandwagon and have a most responsible person for hazardous drug or a most responsible person for sterile compounding, these people do not have to be the same person. You can, the supervising pharmacist or the organization can identify another person to be the most responsible person to head up the non-sterile compounding program. I alluded to it a little bit at the top, but there has been a huge harmonization in language and terminology from the sterile chapter. So they will use those terminologies and those acronyms interchangeably, just to sort of cut down on the confusion. There have been large differences in personnel hygiene and garbing, the requirement of the use of gloves, a focus on hand hygiene, and a more formalized process of garbing. And we would expect that you would incorporate that into very straightforward and explicit policy and procedure for your facility. So this is taking a lot of the secundum artem, a lot of what used to be considered just, along for the ride with practice is no longer that. It's really a formalization, and that's reflected in the revision. Now, this is one of the seismic shifts in the chapter, and that is more guidance in relationship to the use of water in non-sterile preparations. And for those of you that are not aware of it, there is a chapter that a lot of us that don't compound on a very regular basis are not too familiar with, but I would direct you to USP 1231, and that is water for pharmaceutical purposes. For those of you that are familiar with that chapter and get confused about what kind of water should I use when, and I think Mark mentioned it earlier, tap water, not a good choice. Generally speaking, not a good choice. There is a pretty easy to follow decision tree within 1231 that will guide you to the selection of the proper water for non-sterile formulations. So generally speaking, distilled water is no longer the be-all, end-all go-to for those of us that are working in the non-sterile lab. Chances are a lion's share of the non-sterile preparations, you will probably be using sterile water for irrigation is probably a very good choice, or some sort of, as the chapter characterized it, processed water. So please, when you are developing your master formulation documents, when you're looking at, if you're looking at reformulating your current non-sterile menu, please refer to 1231 and assure that you're using the correct water for compounding. This is one of those areas where state authority and the FDA are making lots of citations about the improper use by pharmacists in the compounding process of their selection of water. The chapter has a pretty extensive section on formalized training, on-the-job training, shadowing without return demonstrated learning tools is no longer sufficient training. Simply holding a license and saying I did or I didn't pass part 3 when that was available is no longer good enough. So there needs to be formalized training. There needs to be documentation of that training and record keeping. So please be aware of that, and we'll talk about that a little more in upcoming slides. There's more specificity in the cleaning and sanitizing of the non-sterile compounding area. In the good old days, when Reagan was in the White House, most pharmacists would grab a non-sterile bottle of isopropyl, throw it around, and good enough was good enough. It smelled clean, it looked clean, it was clean. That is no longer good enough, and there needs to be a more specific, more formalized cleaning process. And there are charts and tables in the new chapter that I'll point out to you as we ramble through my collection here for you. This is probably one of the areas where I get the most questions, and that is when you review master formulation records and formalize those documents, making sure that your BUD determinations are correct. Mark spoke a little bit about that 14-day window and what to do as you approach that. There's obviously there is an entire USP chapter on the activity of water, and that would be inside USP 1112. Obviously, if we're talking about water, that points to, for those of you that are guessing, the primary pathway of degradation is hydrolysis. You would be correct, and that's what they're really talking about. So in the chapter, if you're looking at it, there is a lot of information surrounding Table 3. And the magic number when we're talking about the activity of water and non-sterile formulations is you really need to know if it's greater or less than 0.6 as an AW value. There's a lot of information within the chapter. Table 3 is a good jumping off point. The other place is if you go into the USP and you find a monograph for a formulation that you want to use or recreate, there'll be very specific instructions inside the monograph. So take a look at that as you go through and check your formulary and make sure that since there is a formalization of master formula and compounding records, that you have those proper documents in place. And the master formula record and the compounding record are, if you think about it, they are the same document. The difference is that the master formulation record is a template and contains no lots or expirations of the components. The compounding record is the actual record of compounding, or the batch record, and that obviously would contain all of that information and initials and signatures per state law. So, and possibly DEA regulation if you're talking about compounding of a controlled substance. So please be aware of that. And then there's, we're going to talk a little bit about the difference between what constitutes quality assurance versus what is quality control. So as we ramble through, I'll try to clarify all of that. So as far as training must occur before someone is allowed to independently compound. So training cannot be on the job. It cannot be shadowing. It must occur before you allow an individual to compound independently. Training must occur initially, so upon hire or transfer to the department, and then annually thereafter. All of this training must be documented in a training record or an employee file, and at a minimum, the training must cover those 7 areas. So hand hygiene, garbing, cleaning and sanitation, transportation of components, especially if your operation is extensive, or especially for those of us that have satellites that cover a wide area. Measuring and mixing, making sure that the proper devices and the proper amount of accuracy is involved in the measuring and mixing. proper use of equipment and devices. There, depending on the complexity of the equipment that you have in your non-sterile operation, there may be, or there most likely will be, specific policy and procedure, specific training checklist, specific competency-based learning tools to document a return demonstration of especially things like automated pumps, things like undulators, some of the more complicated non-sterile compounding, compounding equipment. Sorry. So once you get beyond the spatula and an ointment slab, you're probably looking at, you're probably looking at more complicated documentation. And, those of you that have met me before, those of you that know me, I'm very proud to be a pharmacist. I think we all do great work, but universally, and myself included, there comes times where our documentation suffers in our rush to provide for a patient, and we need to close those gaps, please. So the documentation of the compounding process itself must stay at our, in front of mind. So let's talk a minute about this formalized training and evaluation. The training must be formalized, it must be documented, and it must contain not only specific operations, but a general understanding of the requirements of the chapter. So how did we get here? Why is 795 important? It's non-sterile. We really can't hurt anybody, can we? So that's frightening enough when you really start to drill into some of the conversations that you have during the training process. An understanding and clear interpretation of SDS sheets and the certificates of analysis that we use to identify bulk substances, active pharmaceutical ingredients, and some of the lab testing that Mark was talking about. If you look at a certificate of analysis from a laboratory and you're going to use that to extend the BUD, especially in the context of creating bigger quantities or batches, especially if those batches are going to be held for any amount of time, was the testing conducted according to USP stability indicating methodology rather than just a simple potency or time point analysis? And I think Mark did a great job earlier. Those are not the same. They're not created equal. So please be aware of that, and please very careful when you go to extend beyond use dating and use this data. And then really making sure that your documents are clear and concise and lead to a greater understanding of each individual's use of policy and procedure and what your expectation is for them in the compounding process. So what's Lou talking about? Why isn't quality assurance and quality control, why aren't those terms interchangeable? In the chapter, as well as in the sterile chapter and in the radio pharmacy chapter, quality assurance is in reference to the system of P&P and oversight that produces consistently quality items. Quality control are the documentation of the results. So QA is the system. Quality control, using the term quality control, is the data. So please be aware of the difference and do not blend those two together, because you'll not only confuse yourself, but it could lead to problems down the road when you're talking to larger groups. So QA systems must establish a system of policing of and adherence to the policy and procedure in place, prevention and detection of errors and other quality problems. My company maintains the largest private database of FDA inspectionary documents outside of the FDA themselves. One of the top 5 consistently over the last 5 years, one of the top 5 citations is failure to thoroughly investigate deviations and detections of errors, whether or not the compounded preps, sterile or non-sterile, were distributed. So it's not just good enough anymore to say, well, we detected a problem or a potential error and we caught it before it left the pharmacy department. Whether it's distributed or not, a root cause or a possible root cause must be sought so that you can go back and feed back into the QA program the data from quality control and make sure that you've done your failure modes analysis and the chances or the likelihood of repeating that error has been brought down to a more reasonable, more manageable possibility. Okay, we're not perfect, but we need to wring out as much risk as we can. So it's not a question of, does the practice of pharmacy contain risk? Yes, it does. We all know that. We deal with it on a daily basis. The question, especially in the context of extemporaneous compounding, whether it's sterile or non-sterile, is whether what we do and by how we do it, are we presenting an unreasonable risk to ourselves and our patients? That's the question that we need to run through our heads constantly. Okay. Evaluation of complaints and adverse effects are part of your QA program. We would suggest that you build into your complaint process and your ADR collection the use of the MedWatch system, the federal MedWatch program, and share that data where and if appropriate. And that will help you achieve the last bullet there, which is appropriate investigations and appropriate corrective action. So the FDA MedWatch program is a really good addition to your arsenal. And for those of you that don't have that as part of your manual, we would recommend that you incorporate that directly into your policy and procedure. Okay, so let's talk a minute about training. The designated person or persons are responsible for creating and implementing your training program. The frequency of training, the process for evaluating competency. The bottom line is you must equip your personnel with the knowledge and training and all of the core skills so that they can successfully perform their tasks. Personnel who compound or have direct oversight should be, again, initially trained, and it must be refreshed at least annually. Depending on the scope and the broad base of your non-sterile compounding program, some organizations are choosing to break the training up into more manageable chunks, and we'll do it on a monthly subject basis or quarterly, as long as all of the areas are covered during that 1-year span. That's fine. This is becoming look at the last bullet. This is becoming more and more interesting in the conversation. More and more state boards are getting involved in this conversation. For personnel that not regularly or do not directly compound and only perform functions such as in-process checks or final verification or simply the dispensing of these compounded non-sterile preps, they also must undergo training, and you must assess at this point, because our state Practice Act does not get too specific in this area. You can really define that in your SOPs, but just because you don't directly compound doesn't mean that you can escape compounding training. Obviously, The New York State Board thinks it is so important that there is a CE requirement, and that's why you poor folks are stuck with me this evening rather than watching your favorite sporting event or catching a rerun of The Big Bang Theory. So let's keep that front of mind or top of mind. All right, let's dance through what does my physical plant need to look like, and maybe more specifically, what does it need not to look like? Your compounding areas, and again, if we're talking about non-hazardous versus hazardous, obviously that must occur in two different places, but a compounding area in general must be designated for use for non-sterile compounding. It doesn't have to be exclusively designated, but when there is non-sterile compounding occurring, it must be cleaned and sanitized before use, and it must only be used for that purpose while compounding is occurring. Okay, so gone are the days where you can leave your nasty coffee cups in the sink and they sit there until tomorrow. If you're using a sink that is designated in the compounding area, all of that material has to be removed before compounding begins. The area must be well lit, and that I would say if the coffee cups are removed, it is more orderly and sanitary. The storage area, where you keep your bulk chemicals, where you keep your API, and this really covers as well where you keep your solid orals and non-sterile dosage forms, must be temperature controlled. I would refer everyone who's listening to USP 659, which establishes controlled room temp, what constitutes refrigerated. And in general, we all know that that range is between 68 and 77 degrees Fahrenheit. Excursions above 81 degrees Fahrenheit for 24 hours are a real problem that could trigger, under both state and federal law, the embargo of your inventory. So you want to have that area temperature controlled. You want to monitor that. You also want to have a contingency plan if for whatever reason you lose control, you lose temperature control of any area that you're using for drug storage. Everything, especially bulk substances and chemicals, must be stored off the floor. So for those of you that are using large containers of things like excipients, base, ointments, and things like that, proper dunnage to keep it off the floor, to keep it neat, organized, and sanitary must be employed. And again, I'm going to talk about water. I talked about 1231, water for pharmaceutical purposes. I talked about 1112, talking about the AW, but hot and cold running water obviously must be readily available. Your sink must be cleaned before use and compounding, no coffee mugs. And then you need to identify when purified water, distilled water, or water that you're creating from reverse osmosis should be used for rinsing the equipment and utensils that you're using, depending on what the final dosage form is going to be and where that final route of administration is going to end up. And all of that should be reflected in not only your policy or procedure, but it should be reflected in the individual master formula documents that you create for each one of these doses. Okay, you need to be that specific because that is part of the release criteria that you or a fellow colleague will be using to do your final check of the preparation. So we want to be specific. Let's talk for a minute about the BUDs for non-sterile. Mark did a great job, and he did talk about mentioning that it is a risk assessment between the chemical and physical stability of the mixture and its ability to suppress microbial overgrowth, right? You must consider the activity of water, the amount of water. Generally, the higher water content in your formulation, the more susceptible it will be to hydrolysis. So we saw early on when 795 was released in its latest form, we saw a move to, especially for things like creams and ointments, we saw a change in the bases to choose bases that were less water-based and more hydrophobic. So that is not a bad idea. And reducing the AW really assists you in the prevention of any kind of proliferation, especially if you're holding these formulations for any amount of time. Okay, so really looking at those, looking at your formulary, and most of you are going to tell me, hey, my hospital, we have a menu of 200 or 300 items in our recipe book. Start with the formulations that you do on a daily basis and work backwards through the more popular items, and eventually you'll get through the entire list. Don't panic. This is one of those cases where, the Boy Scouts used to say, how do you eat an elephant? And the answer to that question is one bite at a time. So just work through and you'll eventually get through the whole formulary. Here is a general table. This is table 3 from 795. So we're looking at 14 days for non-preserved aqueous storage in the refrigerator, and for preserved dosage forms, 35 days as a max. Oral liquids, if you have an AW that's less than 0.6, you can keep them up to 90 days, either controlled room temp or refrigerator, depending on the specific formulation. And other dosage forms, especially if they have a monograph in the USP or you have outside testing, you can take that out to 180 days. But that is pretty much without independent outside lab testing that Mark and I have described this evening. That is really the limit of your non-sterile universe. So gone are the days where you can make a batch of something and hold it for a year. So 180 days is your universe. And I think that's plenty. If we're making stuff that we're holding longer than that, it's really a risk mitigation equation, and you're just entering a zone where you're, by doing what you do the way you do it and pushing the BUD, you're injecting additional risk into the system. So with a monograph, you can take it up to the time the BUD specified in the monograph, and then again, without stability information, you need to really consider not only the formulation, but the container closure system that will house that formulation. And then you need to have the proper USP stability indicating data before you can push stuff beyond what's specified in tables 3 and 4. Okay, so be really careful if you're going that far down the trail. Okay, so here is more of the formality of, again, this is not the days that I alluded to earlier where you just throw a bunch of alcohol on the countertop and you're ready to go. It specifies here the work surfaces, the floors, the walls, the ceilings, and the storage shelving. So the area, it's important to define a compounding perimeter or a defined area for compounding, because it does bring storage shelving and areas like the ceiling into the conversation. So limiting it to an alcove or a room, which is preferable, then some of these cleaning duties become more simplified. They become more well-defined. When we talk about to customers and to colleagues about, the choice of surfaces, the choices of building materials, because of this more formal training process, obviously Your compounding areas can no longer have any kind of carpeting. That's a no-no in the chapter. The ceilings, it's not required that you use hydrophobic sort of clean room tiles, but that's not a bad idea, so that it makes cleaning that much easier. Storage shelving should be neat and orderly. If you're using bins, everything needs to come off to the shelf, and the stuff in the bins needs to be cleaned. The walls don't have to be cleaned except as defined by your policy and procedure, unless there's a possible contamination or when those areas get splashed or are visibly soiled. So be aware of that. However, the floors must be cleaned daily and must be cleaned in between preps if there is a spill or a splash. And then the work surfaces have, just like the direct compounding area in a sterile situation, that is more well-defined, needs to happen at the beginning of each day, at the beginning of the shift when compounding occurs, and then when there's a known spill or contamination and needs to be cleaned in between preparations of different bulk chemical or APIs. So this is just, common sense. Let's talk for a minute about master formula records. So as you are reviewing the BUDs and want to build that back into your recipe binder or your recipe database, these bullets absolutely must be included in the master formula record, the BUD, a final physical description, how, your release criteria, references to support a BUD that is beyond one of the tables in the chapter. That's essential. And then basic things like labeling requirements and other information to clearly describe the compounding process. And your goal is to ensure repeatability from practitioner to practitioner, from shift to shift, and from week to week. You want consistency. It is all about control of the process and control of the final product. So make sure as you look through your recipe book that your master formula records have all of these bullets. We find using a rigid template as a starting point is a really good idea. Okay, so as a way to start bringing this all together, we'll talk about some tangible takeaways from tonight's chat. So be familiar with the chapter itself. For those of you that are consistently compounding, if you haven't actually read the text of 795, shame on you. In the same way, you need to pull down from the state website the relevant parts of the Practice Act and read the documents themselves, okay? Don't take Uncle Lou's word for it. I got the 25 or so slides from tonight's presentation. That's what I'm going to use as my basis of truth. Thank you for the compliment. That is not acceptable. Please go back to the source documents themselves. In addition, please go to the FDA website. There are 27 guidance documents that refer to compounding. There is a specific hospital compounding guidance in the FDA library. Please take a look at that. There is also the broadest, most widely used guidance document by the agency. It is the insanitary conditions guidance for the FDA. Please look at that guidance. It lists 36 specific situations that the agency will take action against a pharmacy, a hospital, or a clinic that is creating or compounding under insanitary conditions. And it starts with the obvious things like insects, rodents, and other vermin Some of the language that the FDA uses is very unique to the FDA, so please read it carefully. You may need to go to the Google to decode some of it, but take the time to do that. The other thing that makes this document so scary is it is not only referring to the direct compounding area, and this is in relation to both sterile and non-sterile compounding. It is not just the direct compounding zones that we're talking about. It also talks about insanitary conditions in areas that are adjacent to your compounding spaces, as well as areas that are adjacent to your facility at large. So please look at that document. It is very interesting. It will change how you practice or how you think about how you practice. Be comfortable with the revised chapter for personnel training and make sure that your training program responds to all of the key areas and the requisites. Please understand the requirements for cleaning and disinfection of your compounding spaces and the tools and utensils that you use. In general, if you can get a disposable vessel or a disposable spatula or pipette, it is probably in your best interest to do that. Otherwise, you will have to have a very strict and very specific cleaning process. And then be very mindful of the beyond use date limits and really your rationale for wanting to push the BUDs. The worst reason for pushing a beyond use date for a preparation that you're creating is the notion that you're ever going to get to a situation where you have zero waste. If you make 4 ounces of a cream and you only use 3 of the 4, you get to that 14-day BUD, dump it and start again. There's no reason to put patients at risk or to risk an ADR or some other unforeseen problem because you're trying to get to zero drug waste. That is just silly. Please don't do that. So just as a quick way to summarize, please focus your compliance efforts to those 3 Ps, right? Training the people, the places, what conditions and what areas are we using to create these preparations, and then the practices. Do I have sufficient policy and procedure to cover the operations, the equipment, and the activities that I do? If not, go out and look for it. If not, go out and create it and make sure that from a policy and procedure point of view, you look at that policy manual, you use it as your basis of training, and you look at it and review it at least annually to make sure that it's not only current and reflects best practices where possible, but it is also really reflective of what's actually going on in the compounding area. Okay? Full compliance to the revised chapter will require a multi-pronged approach, right? Including possible physical plant upgrades, changes to the equipment that you use. So please be aware of that and don't be afraid to incorporate new equipment or new processes where you see fit, okay? Considerations to FDA guidance, the standards of the Controlled Environmental Testing Association, BOP requirements and regulations, and other best in practice guidances from organizations like ASHP will be necessary to close all your gaps. And you're only going to do that if you take a critical eye and do a gap analysis. There are actually formalized gap analysis tools. Everybody is more familiar with sterile gap analysis tools, but there are non-sterile gap analysis tools out there. And please remember, our primary objective is patient safety, right? I think we're all together on that. So with that, I will open it up to questions and refer you to the reading list. And thank you for your time and attention.
Thank you so much for that. We do have a couple of questions in the Q&A box from one individual. Can magic mouthwash be extemporaneously prepared on the spot?
Depending on the complexity of I'll take this two ways. Depending on the complexity of the formulation, it may be able to, it may not, depending on what you guys are putting in there. The other thing is, if the question is coming from a place of the batch, you're dumping a lot of product, then the batch you're making is too big. So it can, but it really depends on the formulation and it depends on your process. I would say from a quality perspective, it's probably better to batch it because that is an institutionally fixed formulation and just have it prepared. And if you're uncomfortable with the BUD that you're assigning now, just cut the BUD back. Can I discuss appropriate API selection? Yes, I can. So there is guidance from both the FDA and the USP. Obviously, the preference is for USP grade active pharmaceutical or bulk ingredients. There is, in the last several years, an additional requirement by FDA that any API, regardless of its USP status, any API that you select to use in a formulation, sterile or non-sterile, be produced in an FDA-inspected facility. And a lot of times that is not apparent. Don't assume that just because 9 other API that you're buying from your supplier are made in a FDA inspected facility, that they all are. So you need to get that certificate of analysis. Sometimes it is not apparent from the C of A document. You need to get an email or a written response from your supplier that it is provided there. And if you're in a situation where what you want to compound or the active ingredient is not available as USP grade, that should give you pause. I think we found out in the latest sort of journey with the GLP-1s and how to get active ingredient to make those preparations that some of this stuff is not always available. And you got to ask hard questions. So just please remember, just because we are highly educated medication specialists, there are times where just because we can put it in a bottle doesn't necessarily mean we want to put it in a bottle. So please be careful of that. The selection of API needs to be covered for your individual facility in a rigid and formalized policy and procedure. There should be a decision tree. API for human use, that's USP grade is the first preference. If you're getting API that are research grade or are tagged with other instructions, that's a problem, or that could be a problem. There are some folks out there that are using things like inositol and other things that are out there available from API suppliers that are not intended for human use. We especially see this in the area of peptides and beyond GLP-1s, but other peptides. So please you're formulating it, you're compounding it, you are responsible. So please be cautious and tread lightly. What else we got here, Gina?
Okay. Can you comment on the BUD that are found in many older references and even in lexicon that are greater than 14 days? How can we assess if the published references meet the standards that may allow greater than 14 days?
Lou, can I jump in on this one?
Mark, help.
This is one of my favorite topics.
Go for it.
Yeah, I'm actually a consultant. Hello, with Lexicomp, and my screen just went blank. I'm a consultant with Lexicomp, and I have been advising them on some of the issues associated with some of their references that are in there. Some of the references are old, and they have issues with the stability. We are evaluating it. I'm working with Lexicom to try to update a lot of these things. They are aware of the issues. You are responsible for the references that you pick. So, anybody hear me?
Yes. Yeah, we can hear you fine.
Okay, I'm sorry, my screen went blank.
No, you're fine. Suffice to say that if you're looking at data and you're trying to make a determination, whether it's sterile or non-sterile, it cannot be anecdotal data. It needs to be, as a minimum standard, it needs to be instrumentally verified. So Mark said early on that he had the benefit of using HPLC. So if you find a paper that either references instrumentally verified using HPLC or gas chrome to back up their data, that would probably be better data than something you pulled out of a journal that was published before Reagan was in the White House or during the Nixon years. Okay, so yeah, don't do that. The other thing is you really got to ask yourself, just because you gave it 6 months dating and you've never had a problem at your institution, you really need to look at those master formula records. You really need to come into compliance with the tables that are in the USP, because you simply just don't want to take it out further than you need to provide good care for your patients. And can I comment on why we shouldn't use distilled water to do non-sterile compounding? It has to do with the testing, and I would refer you to 1231. And it really, because all distilled water is not created equal, because is it RO, is it steam distilled? And it really has to do with the bioburden and the things that are things like endotoxin that and other testing that is not conducted on distilled water. And if you want to maximize the BUDs, even on the tables inside the chapter, you really need to go with purified water that has a package insert and that does have some analysis done to it. Okay, distilled water is identified in the package inserts for things like amoxicillin and other oral antibiotics for reconstitution. But beyond that, put down the gallon of distilled water and come into the 21st century, folks. We know it is a burden. We know it's an increased cost, but it's a matter of you won't know you have a problem until you have a big problem. So don't go to 1231 and look at that decision tree and really make sure that that's reflective in your compounding documents. Okay.
All right, thank you so much, Lou and Mark. There are no more questions in our Q&A field. If anyone has any additional questions that are coming to mind, please feel free to pop them in there while we finish up the last of our housekeeping. So I'm going to close out with a reminder of the CE code for our first session, which was risk for pharmacists and medication for technicians. For our second session by Mr. Diorio, the CE code is sterile for pharmacists and training for technicians. Again, that's sterile for pharmacists and training for technicians. Everyone who is on the call should receive an email in approximately a week with links to claim the CE. If you do not receive the email within a week, you can contact NYSCHP at ceprograms@nyschp.org. And the codes are also in the chat box as well as the email for anyone who didn't write it down. And I see no additional questions, so I just want to thank you both for a really wonderful program, and especially one that's helping us all meet our requirement for licensure. I think this was really beneficial for everyone.